Good. Hi, everyone. Thanks for coming. My name is Solny Adalsteinsson. I'm a staff scientist here at Tyson, and it's my honor to be hosting our seminar speaker today. Before beginning, we want to acknowledge that the Tyson Research Center is located on land stolen from Indigenous peoples who were killed or forcibly removed during the Indian Removal Act of 1830. Tyson is actively committed to the vision of becoming an anti-racist environmental field station and the larger work of making science more equitable, diverse, accessible, and inclusive. We believe that in order to fulfill this mission, it is important to acknowledge the ongoing history of theft, forced removal, and genocide as part of the larger history of colonization and white supremacy, which this field station is built upon. However, simply acknowledging this history is not enough, and we strongly encourage you to visit our website, tyson.wustl.edu, and explore the About tab, where you'll find our social justice commitment, and we invite you all to comment, engage with us, and help hold us accountable as a community to that mission. So I want to thank you all again for attending today, either in person or virtually. And I would like you to invite you all to join us in our celebration of Pride Month this month. For those who will be able to stay on in person, we invite you to enjoy a catered community dinner following the seminar, as well as cake with our community. This week, I'm excited to announce we have a special vegan and gluten-free cake for Pride Month in order to celebrate all the LGBTQIA+ individuals in our community. Now I would like to, so now I would like to introduce our speaker for today. We are so happy to be hosting Dr. Danielle Lee from Southern Illinois University in Edwardsville. Dr. Danielle Lee is a mammologist with an area of study that focuses on rodents, from African giant pouched rats of Tanzania to field mice that occupy environmental gradients across the St. Louis metro area. She is also a writer for the Scientific American Blog Network, and on her Urban Scientist blog, she discusses ecology, biology, and diversity in the scientific community. Her science outreach work focuses on sharing science with underserved groups via social media and outdoor events. And in 2013, she co-founded the National Science and Technology News Service, which aims to increase diverse science news sources across the mainstream media and to bring science to Black audiences. For her years of research and activism, she has been named to the Explorers Club 50 as a National Geographic Emerging Explorer, a TED Fellow, and one of Ebony Magazine's Power 100, and a White House Champion of Change in STEM diversity and access. So huge thanks to Dr. Lee for joining us today, and let's give her a very warm welcome.
Thank you. Let me get situated for you, if you don't mind, because I feel like I mumble a lot anyway. My drawl gets in the way. All right, how y'all doing? Great. All right, let's see if I got situated. Push the button. That button ain't working. Okay, there we are. Okay, so I want to talk to you a little bit, just give an overview, a little bit of the work that I do, both with field mice, little small little rodents that can fit in your hand, up to giant pouch rats that can get about this big, so it's about 2.5 feet. All right, and I'm interested in understanding nuisance rodent ecology. And the reason why I'm interested in that is, one, as I was starting my research career as a master's student, I knew I wanted to do research. I knew I wanted to work with exciting, wild, sexy megafauna, and that just didn't work out. Only a handful of people actually get to do exciting things like work with wolves and lions and stuff like that. But then I also started thinking about even early on as a master's student, which was forever ago, about what my research career could look like. And I started learning about a lot of the complexities, grant funding, paperwork, permits, political turmoil, so you no longer have access to things. And I really wanted to proof my career from those kind of things. And so I really started thinking about, well, how can I work with accessible species, backyard species? So I still wasn't necessarily thinking about working with rodents, but I did find it really attractive to work with species that I knew were right outside my back door. And that's pretty important because as a little girl, I grew up in Memphis, Tennessee. I was always interested in my role models were those wildlife kingdom shows, essentially, and I wanted to do that. And yes, I got a chance to go to Africa and do the safari and actually work in Africa with wildlife, but I never imagined I was going to be working with, smaller wildlife. I still had been romanticizing these big ideas of working with something, big and sexy.
Keep going.
Okay. Yeah. And so I never imagined that I was going to find such an amazing career that was going to take me all over the world. So I got to do all the things I dreamed I would do, but not exactly in a way that I thought I would. So giving you just a moment. Are we good now? All right. All right. It doesn't do it automatically. There we go. And so that's how I came. So working with the giant pouch rat. So that's me in the field, a cornfield, because I work where the animals are, and they magically enough, they like to go places where they know food is guaranteed. Agriculture fields of those. And this is the giant pouch rat. This is one of my fellows that I caught in a medium-sized tomahawk trap, So the size that you would use here locally to catch like possums and raccoons, those are the size I use for the giant pouch rats. And as a result of this research with the giant pouch rat, that's how I became a part of the TED Fellow program. And I gave my very first TED Talk talking about the research that I do with the giant pouch rat. Really amazing creatures. From nose to tip of tail, they can get about 2.5 to 3 feet long. Body size wise, they're about the size of a house cat. And they're amazing because they're able to save lives. They can be successfully trained to do one of two things for sure. They can sniff out and detect landmines by focusing on TNT, the odor signature in TNT. They can find landmines that have been in the ground for decades, up to 6 feet down in the ground. And it can also sniff out the odor signature to determine tuberculosis. So in a spit sample, so they can actually detect that. And it's actually, they're working on right now how to help people in human rescue recovery. That one's still gonna work, but they've been working on that one for at least 10 years. And in the news about a year ago, you probably may have heard about like this rat that won this gold medal. So Moragoro was this amazing pouch rat that had helped to de-escalate and demine parts of Angola. And more recently, the organization that trains these rats actually are in Cambodia and Vietnam. All right. So I studied them because they save lives. And so this rat was talked about in NPR and New York Times, and it was really, really made the news. He only just recently died earlier this year. The pouch rat, these guys can live up to 7 or 8 years in captivity. So they're really long lived. And they can be trained to detect explosives in landmines because they have such a great sense of smell. And I was really fascinated by that. So I joined a neuroethology lab to examine the behavior of pouch rats. And so the nonprofit that trains these rats in Tanzania, they have been successfully training them to do that for about 10 years by the time I joined them in 2012. And how they do it is good old-fashioned operant conditioning, positive reinforcement. And so when they find the mine, so that's a sample one. I'm sorry, I'm losing the mouse. That's not what I meant. The mouse has disappeared on me. Is it a touchscreen computer?
We're seeing it circling the mind.
Yeah. I don't know if I'll be any help, but
It doesn't show up here.
if you maybe just looking on screen, if it's not too weird, because it does show up over
That is so
here.
strange.
That is really weird.
So I gotta go over a little bit more. Nope. All right, I'm just gonna have, all right, let me just do it, roll with it. Take 7. All right, so when they find the landmine, landmines don't normally sit up this high on the ground, as shown in this picture. They're actually way, way below. And when they're pressured by pressure, so it can be it can be a lot of soil on top, a foot, 2, 3, 4, 5 feet of soil on top. And especially over time, because throughout the world, from these wars, some of the most recent ones most of us may be familiar with, if you're Gen X, is the wars that happened throughout Southeast Asia. So the Vietnam War and some of the conflict there were a lot of landmines left behind. And over time, the soil piles up. And what happens is people move back into these areas and then boom, literally boom. So landmines take a lot of precious land out of agriculture, out of road building, access to schools, churches, building lives, and it literally destroyed not just people, but it also disrupts wildlife as well. It's a conservation issue. And so when they can smell it, just that leftover signature from decades before, they'll just attend to that spot and they'll stop. And then the trainers that are nearby make a note on a grid essentially, and then they go back and then they send the big machinery in to essentially blow it up safely. You just have these big tanks that can take a hit, and that's how they demine it. This research was funded, that I do, by the U.S. Department of Defense Army Research Office. They were really, really interested in this because they're like, this is pretty neat. You can use these rats to go detect landmines. They're lighter than dogs, so they don't tend to tip them off. The rats don't attach to the trainer. And so one of the setbacks with using dogs, which are still the standard bearer of landmine detection, to be honest, is that they attach to the handler. And the dog usually only has a working life of about 2, maybe 3 years. And then you have to pay this entire team of the handler and the dog together to go out and do that. These rats don't attend to the handler. So you're able to train lots of teams of people and then send over properly trained rats to a different team anywhere around the world, and then they can hit the ground running doing the work. Here was the problem. This was great, but the organization at the time, they still had a lot of hits and misses. It would take up to a year to train rats successfully, and only about 35% of the rats could pass the test. So that was a lot of investment. And they didn't have reliable breeding. Like, there were no predictability in their breeding numbers. It's not like they were like, we're gonna have 50 rats this year to train, even knowing it was that 35%. They literally can have 5 rats born a year or 100, had nothing. That's because they put the applied science in front of the basic science. They literally didn't do any basic science. And it's because no one on the team was trained to do that. They were all just, occupational psychologists. That was their focus. And so the Army was interested in this for obvious reasons. And they were interested in ways to potentially scale this for not just national security, but potentially homeland security. They're still very interested in that. I was brought on board the team because I'm trained as an ethologist, so super old school animal behavior, where you're hyper-focused on understanding animal behavior as it relates to their natural history. So part of my activism within science communities is understanding their natural history is still critically important, regardless of how sexy and exciting the other science is. None of our science makes sense, especially if you're working with wildlife, if you don't understand what makes them them. And so I was brought on board this team because I had skill set in field biology, field ecology, and working with wild rodents. And I did behavior tests using psychological apparatuses. And I had learned during my dissertation that, oh, these psychological apparatuses don't always work the way that we think they're going to work in a behavioral ecology and a natural history context. So I had that experience of kind of thinking about behavior in these multiple ways, but even though I still had these strong inclinations in natural history. I'm hosted in Tanzania by Sokoine University of Agriculture. And when I came to this neuroethology group, I had my strong background in ethology really did me well. So I'm hosted by the pest management department. This is important. So still telling the story of the pouch rat. The pouch rat came to be this exemplar for saving lives because this university, the pest management department, had pouch rats on campus because they were studying them as crop pests. So you saw me in that big cornfield. Pouch rats, first of all, are really large rats. They build these really huge dens underground, and we strongly suspect that they build these huge, very complex tunnel systems under the ground as well. And we know above ground, when I catch them, they can cover like up to 100+ meters easily, like just like that. And so my trapping grids, when I trap them, so those of you all who might trap, we tend to think of like for small mice, we're trying to do everything every 5 to 10 meters apart, up to 100 meters total. I was trapping in scales, spacing things out 20 meters apart because 10 was just too close. So every 20 meters, I'm spacing out And I go these really, really long transects, essentially until I either hit a house or the street, or I didn't have permission to be there, just to find these rats. And so they're really, really large. They're really, really long-lived. They're really mobile and agile. They can jump from the ground up 6 feet above my head without a running start. They're really, really good climbers. And so I was focusing on that and spending time in Tanzania. I got there excited because I'm like, oh, I'm going to do something nobody else has done. Lesson number one. So spending time in Tanzania also really, really brought me front and center with how much, despite being a Black person from the South, and that's important because I come from descendants of enslaved Africans as well as removed Native Americans. I had what I thought this like very understanding approach. Like I was like, yes, I see the world differently. I do science differently, which is true. But it wasn't until I got to Tanzania and East Africa that I realized how Western I was. I still had a lot of unacknowledged expectations. And one of those things was the fact that I thought I was going to really be doing something. I thought I was really discovering something. What I recognized eventually is that I had an amazing opportunity to work in partnership with local Tanzanians, both at the university and in the local communities. And then I was eventually trusted enough to participate in what's the word I'm looking, in codifying this science for Western, codifying this information for Western science and working alongside them. Yeah, the mouse still doesn't like me. It still has disappeared. All right. So this is Moragoro, Tanzania. This is where I study pouch rats. And you'll note if you look out in the background, you see mountains. That's the Uluguru Mountains. And you see like a town there. That's an urbanized area. And then in the foreground is a rural area of the Shamba. And so I pretty much study pouch rats across that entire environmental gradient, catching rats in an entire environmental gradient. This is my primary research site, especially during my earlier trips to Moragoro, the rural area of the Shambas. And then here's where I study pouch rats primarily now, in addition to the Shambas. This is the town. So this is like the town part. And this particular neighborhood is called Mtawala. It's a working class neighborhood in Moragoro where my trusted research tech lives. So this guy, Mr. Shabani Lutia. And now here I am in this picture taking morphometrics of the pouch rat. So you see it's in this nice little sheath. That's their rip-resistant fabrics of sailboats. They can still bite through that. Not easily, but that's like how we have to sheath them in order to handle them. They are really strong rats. More things I kind of learned along the way of working with this animal is I'd only worked with field mice before. I had done a pretty good job. Pouch rat is really big, almost like the size of this thing, perhaps wider. And some of the things that we don't talk about, the gendered aspect, particularly in science, is hand strength and hand width. You know, I was being recommended to just grab them across. I was unable to do so. Like, we don't talk about that. So a lot of things I learned. So the pouch, I needed the pouch not just to keep them from biting me and keeping me safe, but that was one of the ways I really developed for handling the rat, because I was unable to handle them. And not only that, they're strong as hell, and nobody tells you that. That they literally can bear down and you can't pick them up. Like, they're just really, really, really strong. But I also talk about Shabani, because Shabani is a tech. And so you work with the local techs at the university, and when outside researchers come in, then they work for the university, but then they make this additional money by working with those of us who come abroad. Well, I came to find out that nonprofit organization that I talked about is through the university. It's what we would call here in the States a public-private partnership. They don't call it that, but that's essentially what it is. And it's ran out of Belgium. It's a completely Belgian team. They bring in a lot of Western researchers that run all the research. They hire a lot of local Tanzanians to do all the handling and training proper, but all the scientific expertise was saved for like literally one or two, almost all exclusively come from Europe or the States or New Zealand. And it became abundantly clear to me when I got there, I was the first Black person who wasn't African that they had interacted with professionally, which caused a lot of interesting disruptions, especially in engaging with them. And I came to realize that every rat at that facility, every rat that had ever been trained, Shabani had captured all those wild animals. It was his expertise that they were depending on daily for like finding these animals, pairing animals up for breeding, for all of that. And so because he didn't work there, they just put animals together. But every rat that has ever saved a life, this man is personally responsible. He captured those rats. And I was out in the Shamba, the rural area, catching rats because my earlier experiments were focusing on, can we understand their social organization? Because one of the earlier questions that we got from the organization was, they were like, well, we just kind of put 2 rats together for 2 months and we come back later. And sometimes they have babies, sometimes they kill each other, sometimes they don't. Like, it was just like, I was just like, what? What are you talking about? And as I was spending time like observing their animals, I started noticing a lot of things, which I'll talk about in a moment. But one of the things I really started learning when I learned it was Shabani, who had caught those animals, he finally told me where every single rat that has ever gone through that program came from. Here, his backyard. They were all nuisance rodents that had been removed from his neighborhood and sold for bounty to the organization. And their housing system, as you can see here, it's a lot of like sheds kind of put together, a lot of makeshift homes. There's a lot of like micro stock, like chickens and small things. Plumbing, if you're wealthier, you have some more formalized plumbing. They still they have what we call outhouses and outside showers where everything just kind of, they build not even a septic tank, but just a holding tank. And then when it fills up, they cover it up and they dig a hole somewhere else. The rats are living all throughout here. This is where the rats live. They were all vexing them, getting into their houses or digging underneath the housing, causing some collapses. And so, but I thought that was really important. I thought, I wonder if they have such a hard time, why they have such a low success rate of training animals, why they had such a low success rate of breeding. Not only just because they didn't know much about the basic biology of the animal, because they were nuisance animals to begin with, and like what that means developmentally. And so that really caused me to really, really think it. So in this neighborhood of MG, I started thinking like they live in very, very different environments than the ones in the Shambas. Like they're living a lot more clustered together. This is 3-dimensionally more rich and complex and interesting. More noise out here. There's more light. There's all this stuff that just doesn't exist out in the countryside. And so I really began to rethink my scientific role and purpose there, especially after I learned that's where the rats were coming. And so Shabani started taking me to where the rats came from. So I was literally knocking on people's doors asking, can I catch rats? But here's the thing. I'm a catch and releaser. I was like, and I need to release it when I'm done. I felt really, really awkward about that. And I explained to them what I was doing and why I wanted to know. I wanted to know, I can catch it from here, but I'm really curious to know where do they live here? Like, this is just such a different place. Like when I released them back in the, in the Shambas, there was a hole I could find. I was like, where are these holes? Are they traveling above ground more? Because it was clear to me that the ones in the Shamba were not really above ground very, very much. Are they traveling above ground? Are they accessing the sewer tunnels as if they're like their own dugout tunnels? Like, what are they doing? And so I still can't believe people said yes, and they did. They let me release animals back. It took the same animals that were like giving them problems just for the sake of figuring out where they were. And we were able to figure some of that out. Like, so after I was able to suss a lot of that out, and then it was easier to start removing animals, especially when we started needing animals to bring to the States. I was like, well, I'll take these animals and I'll take those animals and bringing them and, into captivity. But I swear the more we caught, like they never seemed to go anywhere. They were always there. And they were hiding in they were living in plain sight, like in the trash heaps, because they don't have a formal trash system. Like they don't have dumpsters. They just have a heap, and then if it gets too big, they burn it. Well, one of the most popular trash heaps right in front of Shabani's house was actually a successful den. It was there for years and years and years. Like I could still go back to that den and some rat lives there. Haven't found the same rat from year to year, but it is definitely always a place where pouch rats are. And I was able to set a camera up, to kind of record it going in and out, as well as people walking by, and cats and dogs running by, like they're all just living there, just all there together. And so because I spent so much time in Tanzania, in Moragoro, and because I spent so much time in the neighborhoods, I started thinking about it. So everyone in Moragoro knows the story of the pouch rat. They're like, yeah, this rat saves lives. They know all about it. Everyone's really proud of it. But I thought about it, I said, well, what does this research mean to them? Like, who cares if this rat saves lives over there? It vexes us right here. When is this research going to mean anything to us? What can we do with this information? And so I really started thinking about how can we make this, how can we make even natural history important to everybody? In other words, taking the principles of justice where everyone shares costs as well as benefits of research. And I really started thinking about that. Because when or how could this research be of use to them? And it really made me think, like I spent too much time there. I was actually living in community and I was like, I gotta make this work. And so I really started shifting the questions I started asking about rodents from just simply understanding their ecology and natural history, but I really started wanting to understand it from the context along these urban environmental gradients, taking into consideration of studying something that is a nuisance and started cataloging and taking note of people's relationships with these animals, regardless of what it was, and then using that as a starting point for evaluating like what's happening and what could be done more. And for a lot of people, interestingly enough, they weren't interested in exterminating the pouch rat. It's actually not the worst offender of rodents in Tanzania. It's the little small ones that really rape the crops. For the most part, pouch rats are there, and until they're fine with them until they become a problem. And for them, that it means either finally the crops like disappear, or more importantly, if they're out in the fields and suddenly the ground collapses underneath them because the den has gotten so big, or what happens in the more urbanized areas as part of under their house collapses. That's when it becomes a big problem. Now they're also, but it's also socioeconomic. People who are poor and have less sound home structures. So there's still a lot of people who have homes that are made of just literally sticks and mud. Like some people still live like that, or they have chicken coops made out of that. Like that's the problem. The rats can penetrate those things. So like they want to know information about, like they need information to kind of help them bolster their claims for needing better homes, like, or having investments. Like, yes, invest here, but invest in better homes so that the pouch rat can live out there and we can live here. Because they recognize, you know, we have this relationship. There's a historical relationship between the peoples there and the rats there that go back farther than any of us can even imagine. And so it's not about extermination, but it is about, you know, living, you know, in dignity. And I started thinking about those lessons when I moved here to St. Louis back in 2016. And I now apply those lessons here at home because we also have this amazing rule to urban gradient. And I get the opportunity to test out a lot of my ideas that I have for the pouch rats, but a little bit closer to home. And I still work with folks, primarily my students. So here's a few of my undergraduate students here in my lab. And I also, when I'm lucky, able to work with folks in the communities. So for example, these are students from the East St. Louis Upper Bound program. In this picture, we were researching sites, green sites on their campus and near their campus and training them in basic natural history observation and description, which is just basically going out and just identifying like what lives here and taking abiotic measurements, really, really simple stuff like that, which from their teachers' perspectives, they appreciate because it really enriches those lessons and those math skills and science skills and language arts skills, but it's also still real and relevant. And so we start from that place, you know, because, and I'm very honest with them, like I'm interested in these sets of questions, but those sets of questions open up all these opportunities. And they usually start, you know, volunteering, like, well, at my grandmother's house, there's a snake that comes every winter. I was like, yep, that's that snake's house too, whether you know it or not. You know, or they're like, they start literally asking, like, how can I be engaged more? Can I borrow some equipment? Can we come to my yard next time and do this? And so finding opportunities just open up the door for that. All right. And so with my students and with the community, I focus on various field mice species. So jumping mice, they're cute little mean jokers, various paramiscus, deer mice, and even voles. I catch, this year is a really big vole year where we are. All right, so ecologists and evolutionary biologists have been cataloging the phenotypic differences between populations along gradients, between habitats, across distribution ranges for decades. And we know that these incredible phenotypic differences within species occupy different habitats. So starting from that place, I started thinking about like, what does this mean for something like a backyard species that we can look at like Microtozoa pregaster? And so I historically work with prairie voles during my dissertation, and now I'm back to working with them again. And so here's what we know, just looking at that kind of habitat or geographic range, is that prairie voles live pretty much across this swath from Kansas of the United States, and you kind of go straight across over to the eastern seaboard and a little bit north. In Kansas, Lawrence, Kansas is the place where they've been studied. We know that prairie voles are less monogamous and have a lower preference for their familiar mate. Sorry, I pushed the wrong button. In Illinois, Urbana-Champaign, where they've been studied the most, we know that prairie voles are more monogamous and have a greater preference for familiar mate. So the prairie vole is the one that we often use. It's now become a model species for family dynamics and monogamy. It's a rather monogamous, or at least socially monogamous animal. What we also know, so that's the behavioral stuff, but here's some of the physical phenotypic things. There's greater sexual size dimorphism in the Kansas voles, less in the Illinois voles, where they tend to be monogamous. We also know in Kansas that they have weaker alloparenting, because these guys, at least on the Illinois side, they're like super, super communal. They hang out, even though they should have been old enough to go on their own. They hang out with raising younger siblings. They'll even let strangers come through and spend a few nights and move on to the next one. So they have this huge wandering culture until they like kind of establish themselves and find their own mate. So, but Kansas voles, not so much. They have weaker social behavior among non-related conspecifics. They will fight strangers, like, get out of here, I don't know you. They don't stick around and help with their siblings. The opposite is true of the Urbana-Champaign. And that's important for those of us who study these voles, because a prairie vole isn't a prairie vole. Like, they are, but there's these strong behavioral differences based on that. But this is what we also know with the environmental component. There's patchier, drier habitats in Kansas, and they live in lower densities. The opposite is true in Illinois. It's nice, lush green fields, like clover fields, grass fields, lush vegetation, and that lush vegetation supports higher density of voles. And it supports these high density, and they seem to be like lovely with each other because of that. Like, it's a happy, grand green time in Illinois, and it's like dry and scrappy in Kansas. All right. And so I'm just simply narrowing down this scale of observation. I'm still looking at species across environmental gradients, working with whatever field mice that I catch, understanding here locally, that's what we have is the voles, the peromyscus, and occasionally house mice and jumping mice. And so I focus on examining differences in behavior, ecology, and natural history of mice, but essentially what I essentially do the scientific study of country mouse and city mouse. And the same thing can be said for the pouch rats, the city rats, the Shamba versus, the Nji, the town versus the country. And that's essentially what I want to do. And thinking about this, things we already know, we know that land management, how humans occupy and use land and maintain these physical spaces, creates gradients. We know that. And commensal species, like the pouch rat in Tanzania, or even field mice species, which house mice and peromyscus are a little bit, depending on the more country areas, they at least live next to us. We know that field mice species like peromyscus and voles are regarded as nuisance rodents. So they're all a nuisance to people if they get in high densities. But we know that they also can thrive across a continuum. Both pouch rats and these field mice can thrive across this entire continuum. And so land management is the process of managing the use of that. And I'm curious to know, what is it about these different species? How do they adjust? How do they live? What makes them the same and what makes them different across these scaled down environmental gradients? So here are some of my local field research sites. So Southern Illinois University sits on 2,600 acres of land. The buildings are mostly clustered together with much of the acreage in nature preserve and land management. And so our research building is that small little white spot inside that circle. But around it is one of my primary field sites where I have existing grids of trapping field mice. I also study in more ruralized areas. This is so still in Madison County, Illinois, so the same county as the university, Marine, Illinois, only 12 miles away from campus, but a very rural town. And this is one of my other research sites. So essentially one of my colleagues' land. This is still Illinois proper. This is my department chair's property. Like all of that's his. All of that is his. You know, and so it's rural, but they're not farming it. It's just open land. It's farmland all around. And this was actually pretty green. So this is about 15 miles from campus on the outskirts of town. This old mosaic environment from the aerial images, but it's pretty much all natural, mostly unmanaged forest land. So even within a single county, you can get a lot of variability. And here's one more, still in Madison County. So this is only 3.5 miles from the university, right at the Mississippi River. This is land managed by the Army Corps of Engineers, and it includes a 15-year restored prairie right along the Mississippi River floodplain. Sorry. And so all these very different places, you know, you have a lot of different, how the land has been used historically and even more contemporarily. Now, let me tell you a little bit about what I've done. Take a step. So my pouch rat research. I do capture mark releases. I study them across rural to suburban sites. In between the years 2012 and 15, I did intense trapping season, trapping for 2 to 3 months, basically when I'm there, followed by monthly off-season trapping by my crews once I'm gone. And over that 3-year period, there were over 4,000 trap nights. So there was a lot of trap effort. And we had over 127 captures. And here's just an overall description of what we found. We see that in the agricultural areas, the Shamba, we will catch females, we've caught more females than males, like just consistently. So I just caught more females than males. But in the peri-urban site, so that's closer to the university, so on the campus proper, we see that we still catch more females, but not as many. So, so not so much, you know, a trash heap. All right. All right, so one of the things I want to focus out is the number of recaptures. So we caught a lot of females, but we were actually catching females over and over and over again. Catching males more than one time was actually pretty rare. And it was significant. Females were recaptured more than males. But here's some of the background information behind that. Of the males we recaptured repeatedly, they were almost juveniles. They were subadult or smaller juveniles. Large males almost rarely caught them again, and there was always multiple days. So if you caught a young male, you would catch him on consecutive nights. So we would catch these females consecutive nights and could catch them consistently for like weeks, and then all of a sudden she disappears. And then maybe a couple months later, she shows back up. Pairing that with the oral history that I understand and I've learned about the pouch rat is that the females, they live underground. They store a lot of food. All that they store food. They're known for like, they have a year's worth of food is what the local lore is. Not sure if that's true, but they do cache a lot of food. I didn't say this before. They're called pouch rats because they have cheek pouches. So they can fill their cheeks up like hamsters and take it underground. And something important that Shabani shared with me, Shabani has never, and he's been doing this for 30 years almost, he's never caught a baby or a juvenile pouch rat. When pouchies come above ground and they get caught, they're already at the nearly adult weight. So 800 grams or more. Catching something smaller than that, he said, was only time he ever caught something that small is when he caught a female that obviously had birth and put the trap in the same spot. And the next night he caught this little bitty baby, this small baby that was only 300 grams. Pretty clearly that was a baby looking for his mom. And then he said that was the first time he had ever caught one that small or young in all his years of doing it. I have reason to believe that pouchies don't even come above ground until well after weaning. and they don't wean until they're around 700 grams. They can stay underground for months. Mama might come back out and start collecting food and bringing it. And it also looks like the male and females don't live monogamously. They'll tolerate each other very well. And we know that from the breeding that's been done subsequently in the local populations at Cornell University. A male and female will stick together. He'll even brood the young. But what we think is happening is that these males are cruising, going from female to female to female. But staying above ground is dangerous. Basically, they're like little vampires. If the sun comes out, they have to find a safe house and like hide. And they'll duck in there and they'll stay in there all day until the sun comes out. So we also think they're amazingly tolerant of one another when they do come across each other because of that environmental, constraint. Like, okay, we understand you got to hide out for the day and now leave, those types of things. Right. Let me tell you, I'm going to focus on the whitefoot mice of the local mice that we work with. And so looking at foraging activity over preferred and non-preferred habitats. Now, this is looking at the research from my previous master students, Hannah Griffith. She had this amazing contraption where we were using camera traps and essentially open arenas, but they're not enclosed. And so there's holes here in the buckets. So mice can come in and out at will. There's a little dish inside that has bait in it. And when they come in, the camera trap is triggered. And so we baited these open arena buckets with a trail camera from fall 19 to winter 20. And we captured over 151 trap events, camera trap events. We saw that time of day had an impact on visitation rate. In other words, they were mostly busy at night, something we expected. But that's not because these animals, they will come out in the daytime, but they were definitely most active. And they were visiting these stations, so blocks of time. And they were really, really active throughout the night. So starting at dusk, but by the time we got closer to dawn, the activity waned. All right. And this is looking at different habitats. So Peromyscus leucopus, the white-footed mice, really seem to prefer forests. And so at the first farm area where there's like this open pasture land up into the forest, Peromyscus were found throughout that entire range. But they would feed, they had more visits and they would feed earlier in the evening in the forest area than in the grass area. And the morphometric data of these animals show that the animals that lived in the forest tend to be mature and older animals. And the ones that lived out in the grasslands tend to be smaller and presumably younger animals. So we think that there may be some exclusion, like the little ones can't hold on to it or they get driven out into the less preferred habitats. And so looking at the field mice in various species, studying them over 3 different environmental gradients. So now this is looking at some of my SIUE data. So we have old fields, which are just whatever grows, grows. Brown fields, places where there's been some sort of construction or potential contamination, but it's being grown over, but it's potentially contaminated, as well as restored prairies. So that Army Corps of Engineer land with different surrounding areas, everything from rural to peri-urban. And from 17 to 19, we had intense trapping, 2 to 3 months, followed by monthly off-season trapping. We were active trapping primarily from March to September, and then rotating between the field sites every 1 to 2 weeks. Cataloging nearly 4,000 trap nights over that period of time, and we caught 189 animals. And so this is preliminary capture mark recapture data, but the descriptive numbers. But what are we seeing in these different landscapes? We're seeing that these old fields tend to support a lot of animals. We have a higher number of catches in these old fields compared to the brown fields or even the restored prairies. But the surrounding area, even though it's not significantly different, we do think there's an interesting story being told here. In other words, it's not just a simple habitat where the mice occupy, but what's happening around that habitat may also be contributing to it, either a supplement of resources or interfering with resources, interfering with dispersal routes. All of that may be influencing, how these animals live and survive and thrive in a particular area. So whether a place is rural or peri-urban, in other words, what supplements or resources that are available in the surrounding area. Just focusing on voles, the prairie voles, we were catching more voles in restored prairie compared to the old field and brown fields. and we were catching more Peromyscus in old fields, interestingly enough. So even though these animals can every place we catch them, you can find they all can live there. We were seeing some different things happening. And I think the jumping mice were telling the most interesting story. So first of all, these guys are super, super sensitive to disturbance. I've literally lost them I've lost them for like years. Like they'll disappear in the data set and then all of a sudden we'll trap after a year or two of never seeing any, they're there. So we found that the jumping mice were in the brown fields, but it wasn't just so much the brown fields, they were in the part of the fields that were closer to the edge of the forest where there were a lot of really, really tall grasses. So really, really tall grasses. So here's the connection. What I'm noticing is that regardless of the size of the animal, the type of animal, these animals have spatial preference and they have these patterns. Pouchies, we definitely saw strong sex differences in recapture. What we think is happening is that females have a home and males roam. And so the females stay put. They establish a home and they stay there with their babies. Those babies don't wean until they're around 700 grams. And we know that supplemented by lab studies. That's when they finally stop completely suckling. And even though they can eat solid food starting as early as 2 weeks when their eyes are closed, they don't really seem to demonstrate the behavioral tendencies of dispersal until they get closer to 4 or 5 months. And then based on local lore, they may not even be coming above ground for another 2 or 3 months after that. So mamas stay put, males move. And that's why there's site fidelity in females. Among field mice, some species are caught more often in certain places. Ecologically, as you would expect, different species are occupying specific places and spaces over the observed period of time. So you see these patterns. But taking a closer look, we think these space use patterns make sense according to the life sense and natural history of that species. So for example, female pouchies, their site fidelity, male roamers. White-footed mice, they can't occupy most habitats. But we tend to, throughout our study areas, we found that, but they were more active at night and in forested areas. So there's a preference not just of particular habitats, but perhaps even there's a temporal component to their preferences too. Prairie voles also can occupy many types of habitat, but we tend to find them more open in grasslands where there's lower incidence or frequency of disturbances. And now that we're really pulling all this data in, we found more prairie voles than the other animals overall. So like they tend to be the most abundant. But they love to be out in the open sunny areas. Jumping mice, we only find these jumping mice in the riparian ridges. In other words, near the forest, and we have these ephemeral pools. So they tend to be near there where there's very tall grasses, like as tall as me. Forbs with lots of vegetative biodiversity. That's the other thing that's different. It's not just simply lots of grasses and sedges, but it's a little bit of everything. I don't know my plants, but it's the kind that have things that stick to you. Everything looks like pretend wheat, but it's different types of pretend wheat, and I don't know what they are. But lots of those different plants, and they experience low disturbance and intensity and frequencies. And a mow in those areas, or disturbance in those areas, we can lose those jumping mice altogether for a season or two. And so ecologically, generally with different species, they occupy different places and spaces and time. Here's my big end story. In other words, natural history matters. In other words, it's the foundation of applied science. And when I learned that, I knew that, but I really got a chance to rediscover that with the pouchies specifically. Even if we want to use them innovatively, we still got to know the basics. But here's where I really like to focus, is reminding people that the animals we study have deep evolutionary and ecological histories that shape them. And just even if we can't successfully raise them in laboratory settings, whether it's for a period of time or in perpetuity in captivity, that history doesn't go away. That stuff still sticks with them. Those behavioral tendencies, those preferences, whether known or not, still really stick to them. Those either temporal patterns or those spatial preferences, those things still exist. And it really influences our science. Types of questions we ask, the types of apparatuses we may expose them to kind of get data from them. And a lot of times, especially if you're working with truly wild species, it can really frustrate our ability to find patterns, because when we're working with wild animals, even a couple generations from the wild, they're still reacted to it in very, from my perspective, very broad strokes. And so we don't see a lot of variability necessarily. And so sometimes the trick is to look at the variability and not necessarily the data itself to see where the story may be at. Or what I'm learning is we sometimes, even though we're looking at this particular focal point, we often need to scale back to see what's happening around to see how that influences us. As I tell students all the time when they're learning ecology and they ask me a question, what's the answer to that? I said, well, the answer in ecology is always the same. It depends. And what I like to remind them is that depends. We have to start focusing on that, like really start paying attention to what's happening, in the environment beyond just that particular focal, because place, space, and time really, really matter. And I'll just say, here's the thanks to all the folks I've worked with. So my postdoc associates, my Tanzanian associates, and then of course, more recently here locally, the university that I'm at, SIUE. And I'm available for any questions.
I have a question. With your now understanding the pouchies and like the females have a home and the males roam, do you have an idea of what might be going on when the folks were trying, they threw some pouchies together and either they all killed each other or sometimes you got babies and sometimes you didn't. What do you think might have been going on there?
All right, so the question is, just want to make sure the folks can hear it, is now that I have a better idea of some of their natural behavior and natural history, what might have been going on reproductively at the center. Yeah, so what I don't talk about here is that we learned. So this species is also interesting. The females' vaginas don't formally open until almost a year of age. They were putting immature females in with males that, or they just weren't ready. I know you're like, so there's not, it's closed. It's not, it's just all skin. It's just skin. There's nothing. The paper's finally coming out with working in concert. So they have super delayed vaginal patency in this species. And that, I think I was the first formally to recognize that because the people who work with them didn't notice it. And here's the story of how I discovered it. I was missexing animals in the field because it was closed. And they even have that little line, that little perineum line. And I was just like, yeah, that's a boy. And Shabani was like, no, I was like, oh yeah, that's a boy. He's like, no, it's not. I was like, yes. We were speaking through, like, I barely speak Swahili and he barely speaks English. But he was explaining to me, he's like, no, that's brother, that's not sister. I'm like, no. I was like looking at it. And sure enough, I finally took out the measurement and did the anal genital distance. Sure enough, like that part we were able, like, this is where good old fashioned regular stuff, the stuff you learn in natural history, taking the measurements. AGD comes up every time. Females have a much shorter anal genital distance consistently than males, but we're also seeing variability like you expect to see. Like some females are a little longer than others, and some males are a little shorter than some, but for the most part, it's feminized if it's short and it's masculinized if it's long. I think they were putting immature females or females that were completely not in estrus in there with males, and that's what caused the problem. But I really think most of it was immature because my time there in 2013, I actually discovered about half of their females in their breeding colony were not patent. They just weren't patent at all. But that was not one of their prerequisites. I was like, that should be one of your prerequisites. They should be patent females before you pair with males. And that was that's even before we could suss out their estrus cycle, which we still can. Yeah. Another question? Yes.
Do you have any sense of the size and structure of their burrows?
A little bit, yeah. Do I have any sense of size and structure of the birds? Yes, I do have some preliminary drawings on that. So another interesting thing. So yeah, we were able to use the endoscope. So one of the advantages of being connected with Nat Geo, I was able to get that super expensive little burrowing endoscope. We could put it in there. So yep. So they are interesting. They're not that much different, it seems, than the lesser pouch rat. where it's essentially like a big hole under the ground, and it's somewhat star shaped once you get in there. So they have a big burrow in the middle, and then they have like these, sometimes they open up really big, like really, really big caves inside. And then they'll kind of have a little area for food and a little area for latrine. But what I thought was interesting is they have a lot of blind alleys. So you have one that it ducks in and then it'll just go out. There's a lot of blind alleys. And so what the other kind of interesting sex thing we found in 2018 and 19, I was focusing in the city, so near Shabani's house. Everything around Shabani's house we kept catching was males, like all males. We only caught one female, and we were hardly catching these males consistently. And so we suspect, and they were like one or two big males, but a lot of young males. And we think that the males, especially in the cities, they can tolerate higher densities, and they live in like these little, not quite a colony, but like these little bachelor groups, or at least spatially, it's like, this is the bachelor area. And they just rip and run. And we saw one night, we'll call him Red because we dusted him with red powder. So that's how I was able to do this. I did a lot of the fluorescent powder. We found a male that we caught and we released him. It was getting late in the day. And that's how we realized they'll just hide out. We found a super shallow dugout hole that he dug out. Just a simple little U-turn. He dug out and he stayed there during the day. We could follow it to there, intense red, and then he left and didn't come back. So they're doing so they have a lot of like little blind hidey holes too, where they'll just hang tight and then they'll leave the next day. But they'll also take advantage of most anything. Like one of the more interesting ones we found actually at the Shamba, we found this huge, like it was huge. It looked like the size of a room underneath. And it was like a collapsed tunnel or something. And they took advantage of it. And it was big, like it was big in there. And we had tracked at least two animals. We tracked a male and female there of size, because it was a pink and a blue. Interestingly, the female was powdered blue and the male was powdered pink, because that's the color powder I put on them. And we found it was evidence of them in there together. Like they obviously had occupied this nest at the same time, but then there was no sign of them. And we soon found out why through the camera, because driver ants had taken over. So driver ants are related to army ants, which are horrible, they're the devil. I will abandon entire field sites if I see one, because they will pick something down to the bone. And so that's the other thing. So now like, so one of the things we're not sure of, we saw with the males is like they obviously not staying in the same place every night, because we could follow their track. They were moving around. They seemed to be tolerant. And we also, perhaps, this was way too early to tell, but maybe some other natural history factors like driver ants that kind of come, stay for a night or two and leave, other little predators or predator animals like that is a reason why they might need to like bug out and have someplace else to stay safely for a while, from place to place. Another question? Yes.
We have a YouTube question from James Traeger, who says, really interesting talk. I am pleased to learn how cool these murine animals are. Was there any response or relationship to prescribed fire in the restoration period?
Oh, I'm guessing he means for the field mice, for prescribed. I don't know. I'm gonna be honest. I don't know. We do some burning on campus, but I haven't trapped in the places where we burn. So I don't know, and not gonna. So I'm not interested in studying in the places where they burn. But that would be interesting. Maybe in a few years, I feel like, so right now the disturbance I mostly focus on now is mowing. Like that's my main disturbance, because that ain't even predictable. Getting them to mow, but fire is really, really hard. Like they can have a prescribed fire plan and then it'll get nixed. And then they're like, okay, we're not gonna burn. I'm like, but we really needed that burn this year. So I don't know. Yeah.
Probably time for one more.
Okay.
Have you noticed a difference, or do you know anything about the difference in the diet of the urban rodents versus more rural rodents?
I have not been studying that. Though I do know with the pouchies, I can tell you pouchies because they will void their pouch sometimes for you. They'll spit their pouch out. They will, the urban ones are eating whatever you find in the trash. They eat a little bit of everything. And the rural ones are mostly harvesting grain and local seeds and nuts. But also their local name, one of their local names is Little Thief. So they will, and that was also one of the interesting things we found inside the burrows. There were literally things in there. And he was like, wait, I was missing that. They will take stuff, they'll harvest things and they'll take them into their burrows. So it was evidence of trinkets. The other thing that I thought was the most fascinating, I will say this, I know you didn't say this about food ecology, but burrow building ecology between the urban and the rural one, it's interesting, I'm saying it, realizing this at the same time. In the rural area, in the urban area where they building out of trash, the walls, they use wrappers and trash, almost like spackling in the wall. So if you've ever noticed bird nests that sometimes use plastic, and I'm sure some bird people, I'm sure there's an urban difference, like how much they use recruit trash for nests versus not, if that's a thing. I think that I definitely see that, like, though it's like candy wrappers and plastic trash bags and then woven plastic. Lots of that, not just inside the burrow, but throughout the walls. Like they use it as construction material. It's like spackle all throughout. So their diet, they eat what they get their hands on, I can tell you that. And so, yeah, it's a difference based on that, but I haven't like done any formal things like scat analysis, and other than when they give up their pouch, which they are loath to do. But the only time they won't bite you is when they don't want to give up their pouch. But yeah, I've noticed that the pouch release of the rural ones and the urban ones, they're different. In the mountainous regions where they grow coffee, they do eat a lot of coffee.
Wait, so are they like from caffeine?
Like the coffee beans. I don't think is it the same when it's still the wild plants like that? I don't know. But they eat a lot of those little coffee beans. Like they are so in the north region and the mountain regions, they're a pest for the coffee farmers. Yeah. They eat those little berries. Yeah.
So this might be the most important question to me, since we've already gone the caffeine route, but are the pouch rats any way related to the rodents of unusual size?
They are one of the, yes, they're part of that, but phonology, so there's those, they're over here, they're the smaller group, they're the lesser group. Interestingly enough, they're not the biggest rodent out there. I've caught a cane rat. A cane rat is even bigger. Cane rats can get up to 5 kg because that's what my persona said when I weighed it. And it tanked out. Boom, yeah. Cane rats are even bigger. So of the rodents of unusual sizes, they're the most diminutive of them. They are, which is still pretty small at 2 to 3 kg. Yes.
Do you know about how big the baby fishes are?
When they're born? Yes, I do know that because I had some born on me. It was awesome. So when they're born at between 60 and 100 grams. And they grow really, really fast. So we were able to really suss out, and that was all quick observational stuff. They wean at around 700 grams. Like 700 is when they really start independent. But there is big, like 700 grams is about this big. So imagine a regular rat, an adult regular rat, that's a baby. That's a baby pouchy. And I would, as they start getting bigger, I would see mama nursing. I'm like, which one's the mama? Because they get really big. So 500 grams is still a nice size rat. And females become mature at right around a kilogram. So females can start having babies as young as like, when it is young, well, as small as like 900 to, so 0.9 to 1.1. So my mean weight of mature females was about 1.1 in the field. And so imagine 700, you got a 700 gram baby nursing on you and they all over you. So they're really big, even though they're that size. Yeah, so they seem to be really big. They have some really delayed maturity. And she's not pushing them off. She's laid back and they're just nursing just fine. And I remember just watching them to see how long they would stay before the mom showing disinterest. She never really put them off. Like they still will stay nestled and snuggled together up to about 3 months. But they reached, I was also feeding them a lot because I think that's what I do to show, love. I was always feeding them things. But yeah, they would stay just fine in that little, in the enclosure. But yeah, around 700 grams is when she seems to be less interested, but they'll stay brooding and snuggling together. Yeah. We're over.
One more, let's do it, and then we'll
Okay.
wrap it.
Yeah.
So between the urban and rural couch rats, when capturing them, did they have any different reactions to humans? Like,
Yes, they hate us all. No, what? I feel like on that, just some animals, I think that's where the individual differences came. Like I had some animals who were completely freaked out. And they would like try to run and they kept hitting their head on the cage and hurting themselves. And I saw that in the rural versus the urban areas. Though I'm glad you said it because one of the things, I haven't been able to go back, but one of the things, a simple way to evaluate that is do a simple test of essentially bolt distance or approach distance. Really, really easy. But I don't want to do it in the cage because they'll hurt themselves. So I got to figure out how to, I don't know, put them on a leash or something, or tether, and then just see how close I can get before they bolt. Or because I don't want to, because they really can't hurt themselves. Like they have these scars. But I never did that because when I catch them, I was just focusing on catching them. I wasn't focusing on like, all right, now measure me. 1 step, 2 steps. But then some would just be completely chill. Like some we can get in the bag really easy, like load them into the bag really easy. And once they were sheathed, they would chill. Others the whole time was the whole time. Some were like, okay, I'm just get this over with, and others it varied by individuals. But from my memory, I don't remember any environmental differences in their tendency to be like nasty. No, that would be, I would like to see that. I'm gonna write that down. Thank you. All right, I guess that's officially it. Thank you.